This protocol employs super-resolution microscopy, specifically direct stochastic optical reconstruction microscopy, also known as dSTORM, to bypass the diffraction limit and to visualize EVs with nanometer precision in 3 dimensions.
One notable advantage of dSTORM is its ability to directly visualize particles beneath the diffraction limit of light, without damaging steps that alter the biochemical nature of the EV.
Many evolutionary distinct viruses employ EV signaling. Therefore, dSTORM can be employed to characterize EVs for disease biomarkers and progression, as well as to visualize individual virus particles such as SARS-CoV-2.
Begin by placing affinity-purified extracellular vesicles, or EVs, onto glass-bottom microslide 8-well plates in a total volume of 200 microliters and allow them to adhere to the surface overnight at 4 degrees Celsius. Without removing the existing solution from the 8-well plate, fix EVs onto the plates by adding 200 microliters of 4% paraformaldehyde in 1x PBS to the EV-containing solution in each well, and allow the plates to incubate for 30 minutes at room temperature. Carefully remove paraformaldehyde and excess solution with a micropipette to not disturb the EVs. Wash the EV with 1x PBS to remove excess paraformaldehyde. Perform the wash procedure 3 times. Remove excess 1x PBS. Prepare 250 microliters of dSTORM B-cubed buffer solution per sample by creating a solution of 5 millimolar protocatechuate dioxygenase diluted in imaging buffer as per the